Hi everyone, welcome to my poster presentation. My name is Phong. I'm a PhD student at Institute of Computational Biology, Helmholtz, Munich. Today I'm going to present a project called an integrative and interpretable multimodal machine learning framework reviews pathological signatures of incident DSPN. As you can see, the subject of this study is DSPN. We want to predict the incidence of DSPN, so basically we have two time points, F4 and FF4. Yeah, so F4 and FF4, and what we're trying to do is to build a multimodal model that can predict the probabilities of a healthy person to develop DSPN after 6 to 8 years. So here we have 7 different data types with different number of features, and we also, regarding the sample size, we have different numbers of samples for each of the data type, and they also overlap each other in a complicated way. So what we're trying to do is to extract a subset of samples that have all of the features for the training, and the remaining samples for each data type was reserved for the feature selection. So the project, the analysis pipeline was divided into two main parts. The first part is the feature selection, where I try to select the most important features for each data type for the training, where in the machine learning training and prediction, I try to build the best performing models, and then also extract the most important features for the prediction. So as I said before, I extract a subset of samples that have all of the features for the training process, and for each data type the remaining samples were used for the feature selection. So basically I have 100 stratified resamplings. So for molecular data, I did differential expression analysis followed by gene set enrichment analysis to extract a set of leading edge molecules that drive the enrichment of relevant signaling pathways. And for clinical data, I did elastic net followed by ranking, extraction of the feature importance ranking to select the most relevant features. And at the end, I use a feature rank aggregation algorithm to aggregate all of the feature lists together into a single list for each data type, and use that for the machine learning part. So for the machine learning part, basically I try to implement the forward feature selection algorithm. So first I start with single view model. So I train each of the data type models separately, and then evaluate the performance using 5-fold cross-validation to select the best median performance among the cross-validation results. And then, for example, if I get genomics, and then I test the genomics model on left out samples, and then I try to add more features to the genomics model by also doing 5-fold cross-validation to test all of the possible combination, including genomics, and then keep going and going in this loop until I add all of the features to the models. And then at the end, I have all of the performance results, so I try to evaluate to see at which complexity of the model we have the best performance. And also I look at the feature space to extract the most important features to analyze, and their functions, and also the association to the mechanism of the disease. So this is showing the procedure of the feature selection set for the molecular data types. So basically I use gene set enrichment analysis to select the functionally relevant features with lower effect size. The thing with many metabolic diseases is that they usually involve a lot of lower effect size features, which if we access them individually, they might not play a major role in the prediction of the phenotype because they have very low effect size. But when we combine them together, the combined effect would be significantly higher, and they might play some major role in the prediction. So the idea is that using gene set enrichment analysis, I do not only prioritize the high effect size features, but also I take into account the low effect size one and also when we combine them everything together, we might achieve a higher prediction performance. So that's the idea. So basically, after pre-processing, I did differential expression analysis followed by gene set enrichment analysis to extract a significant list of the signaling pathways for the phenotype of interest. And then after 100 stratified resamplings, I got like 100 lists of significant gene set. So I try to aggregate them together using robust rank aggregation to get a single list of the significant signaling pathways or gene sets. And then I did gene set enrichment analysis again on the whole feature selection set for each data type, but only focus on this significant gene set list. And then using this result, I could extract the final list of leading edge molecules. Regarding the annotation for the gene set enrichment analysis, I use a reactome annotation, which is more user-friendly and more flexible compared to other database. So this is just a snapshot of all of the signaling pathways group that I use in the reactome database for our analysis. Yeah, so this is the result of the prediction for incident DSPN. So basically at first I forced the algorithm to choose only clinical data as a starting point. And then in the next iteration, I let the algorithm choose what is the best addition at each of the data of the model complexity to add to the model. For example, in the next iteration, at the 2 dataset level, the algorithm try to add mostly the O-link features followed by the metabolomics features, and so on. It will keep adding until it add all of the 7 datasets into the models. So this is the testing result of the incident DSPN prediction. The y-axis is the AUC value of the testing, and the x-axis is a different complexity of the data of the model. You can see that when we add more and more features, molecular features, to the clinical model, we can increase the performance drastically, and the performance tend to be saturated at 3 or 4 dataset complexity. Yeah, so here we can see that most of the incident cases have higher prediction probability compared to the control, which show the effectiveness of our models. Yeah, and there's a consistency between the t statistics of the important features in the training set compared to the feature selection set, which show also the effectiveness of our model. Yeah, when I use the most important features according to our model to do the PCA on the whole training set, these features can segregate the cases from the control nicely, as you can see in this plot. And when I stratify the prediction probability of into individual samples, you can see that most of the cases concentrated on the right side of this plot, where we have higher prediction probabilities. And when we look at the normalized values of all of the most important features according to our prediction result, you can see that most of the O-link or inflammatory proteins have higher values in the cases compared to the control. The effect in other features are not really pronounced, but we can see some patterns here. For example, for caprate metabolite, we have higher values in the control compared to the case, which show the downregulation of this metabolite. And for example, people with lower physical activities tend to develop DSPN in the future. So this showed the result of gene set enrichment analysis. So here all the nodes are the features that I use for the training process. So this is the result after the feature selection set, where I use gene set enrichment analysis to select the leading edge molecules. So each nodes here are the leading edge molecules. and the edges are the number of gene sets that are shared between them. So you can see that, and also here the highlighted nodes are the most important features after the machine learning part, and all the rest of the features are in the background with the gray color. So you can see that most of the important features are connected to each other in the center here, which we can use to extract a story of the mechanism of the SPN later on. And this is a few examples of the consistent signaling pathways that are either upregulated or downregulated in both feature selection set and training set. For example, we have here the SUMO E3 ligase simulation target protein, which was downregulated observed in the transcriptomics data, and the two pathways in the middle here, defective SLC27A4 premature causes premature syndrome or IBS, and the transport of fatty acids, they are both downregulated significantly observed by the metabolomic data. And most importantly, the chemokine receptors bind chemokines pathways here are significantly upregulated observed by the proteomics data. So all of the pathways here are consistent between both datasets. They are relevant to the SPN according to several studies, previous studies to study the mechanism of the SPN, and also they are actionable signaling pathways, which means that we can study this pathway and use some medication or treatments to target this pathway to improve the performance of the patients. So in summary, we successfully built a multimodality model to stratify incident DSPN cases from the controls. And using the model, we identify actionable signatures of incident DSPN, and we can prove that the utility of molecular data obtained from the whole blood. However, there's one limitation of this method, in which we rely totally on the available biological annotations. So at the beginning, we have to filter out quite a few features which did not have enough information in terms of function. So that's the end of my talk. Thank you for listening, and feel free to drop me a line if you have any question. Thank you.